Hello, welcome to this event. We are very happy and very grateful for the opportunity to contribute to this humble day celebration. We thank the organizers and the IDS for this amazing initiative. And just introducing very quickly, this event is organized like a symposium on distance-decay models for the analysis of beta diversity. In our group, we are mostly interested in macroecological questions, trying to understand why biodiversity is organized in the way it is at the large-scale levels, like continental and global scales. And we use mostly beta diversity approaches because we think they could be useful to provide new insights about how biodiversity is created and maintained. As I said, we have 4 talks from all of them by members of my group. In first place, Carola Gómez-Rodríguez would introduce some general ideas about distance-decay patterns, and will show several papers we have produced in our group, trying to use creative ways to understand how communities change from place to place, using phylogenetic scaling, for example, or comparing a lot of groups to understand dispersal limitation, or using genetic information to understand how very restricted species with very poor dispersal ability expand their ranges in the long term. Thereafter, Sara Martínez-Santalla would introduce a new method to assess the significance of distance-decay models. We don't have a very good alternative now, other than the Mantel test, which is a linear model. And thereafter, Ramiro Martín-Devasa would introduce a new function, the compare function, which is very useful in some circumstances. For example, when species have very wide distributions and you need to fit a sigmoid curve instead of a monotonic curve. And at the end, I would again focus on distance-decay models, but from a different perspective, trying to see if we can see different things, assessing the degree of anisotropy between the anisotropy of community turnover, and how we can use this to the degree of equilibrium between species and clans, for example. So I hope you will enjoy this symposium, and as I said, welcome. Carola will start with the first.
Good morning. My name is Carola Gómez-Rodríguez, and today I'm going to tell you how we have been using distance-decay models to assess historical dispersal limitations. So just let me share my screen. Here we go. Okay. Well, today we will be talking a lot about distance-decay patterns, which you are probably familiar with, because it's kind of the first law of geography, but applied to biological communities, in the sense that it just means that two biotic assemblages, two communities, will be more similar the closer they are. And you will see a decrease in the similarity of biological communities. That means in the number of shared species and the number of species that they have in common, that with this spatial distance. This pattern is well known. It has been well studied, as we can see by the number of citations to the conceptual framework by Nicola and White. One of the reasons many people use it is because it is a useful quantitative description. And the fact that we can easily quantify this pattern with a slope or an intercept allows us to compare it between regions or between different taxa. So for those of you who have not studied this pattern before, I will explain it briefly. So what we mean with the distance decay of biotic similarity is that if we represent biological similarity against spatial distance, we will observe that it decays, like there is a trend, a decrease, because two localities that are close to each other will have more species in common than two localities that are farther apart, as we can see here in the case of locality S1 and S3. But as you can imagine, two localities that are close to each other not only have a species in common, they have many other things in common, like the habitat, the climate, other factors that might be also affecting the biological communities that we are studying. And this is why when we are using distance decay models, we have to be fully aware that there are different processes that might be driving this pattern. Basically those related to niche processes like, I'll look into this again, climatic distance, and also the ones that are more like geographic structures, spatial structure driven by dispersal processes. As I have said at the beginning, it's very useful because we can quantify it. That means that we can estimate parameters from this pattern, like the slope, the rate at which assembly similarity decreases with distance, or the intercept, that is the similarity at the closest sampling localities. This is easy to see just with, if we compare two distance decay patterns. In the blue one, we have a steep slope. We can see that when distances are quite long, spatial distance, they are quite long, the similarity is low, while this is not the case in the brown one. And we have high community similarity at large distances. And if we think about dispersal processes, we can easily figure out or easily see that if those flatter slopes are more likely to be seen in a species that can disperse well, with a strong dispersal ability, because if two localities are farther apart and they are sharing the same species, we need the species to have been able to reach there, right? In the case of intercept, what we are talking about is this similarity at the closest distance. So when two localities that are very close, do they have similar species in common, or do they have the same species in common, or there is quite turnover even at the closest distances? So with these things in mind, what we have been doing, what we have done these years is use the distance decay models and patterns to assess historical dispersal limitation in different taxa. What we mean with historical dispersal limitation is that not so long ago, but 20,000 years ago, the northern latitudes were covered by an ice sheet, and since then the taxa that were restricted, the ranges were restricted to the south of Europe or the south of North America. They had to recolonize those northern latitudes. So what we are trying to assess is to which extent they have been able to expand their range to those localities that now might have this suitable climate, suitable habitats, but they are still the way to get there. So this, we are interested in this recolonization lag. Why are we interested? Well, because it's interesting per se, but also it has a clear applied interest, and it is that we are all wondering what it is going to happen with biodiversity in the near future with this climate change, and we have a great natural experiment. So if we understand what has happened in the past, to which extent biodiversity or these species have been able to track their climate, we can make educated guess about how they will be able to cope with it is coming, right? So I'm talking about biodiversity, and we talk about biodiversity, we have to talk about biodiversity. We cannot forget invertebrates. We know much less about their dispersal abilities than we know about other taxa like vertebrates or even plants. Well, so they are a huge fraction of biodiversity. We know very little about their ability to cope with what it is coming, but they are also quite convenient for this type of exercise. They have been for us because some invertebrate groups, like arthropods, have attribute morphological characteristics that can be related to their dispersal ability. So if we are able to find signatures in the biographic pattern that we attribute to this dispersal, we can relate to this dispersal or morphological traits related with this dispersal ability. If we identify this signature as the result of this incomplete recolonization and this historical dispersal limitation, then we can try to see if in other invertebrates or in other taxa in general, we see the same signatures, because those could be indicative of strong dispersal limitation. So today I'm going to talk about 3 studies, the distance decay patterns of beetles in Europe, also how we have used distance decay models in the context of phylogenetic scaling below the species level, and finally distance decay, but distance decay in the sense in genetic structure, because in this case, even if it's the same, we are looking at an increase instead of a decrease. So in the case of Europe, we have observed that if you divide Europe into the territories that have been recolonized from the ones that have been mostly refugee or like north and south of Europe, in some particular beetle taxa, we had observed that the distance decay patterns in the north were different from the ones in the south. So we were wondering if this could be like an evidence of this dispersal limitation in this taxa. We tested it with many beetle groups. Our hypotheses were that the distance decay patterns in the north would be flatter as a result of incomplete postglacial recolonization, because the species in the north are the good dispersers. That also the north-south difference in the distance decay pattern would be related to the north-south difference in the species richness, because they would be the result of the same process, this incomplete postglacial recolonization. And we also hope that we could find stronger evidence that the difference in the distance decay patterns were actually related to the dispersal ability of the taxon. So good dispersers would be the ones which would have more similar distance decay patterns in the north and in the south. So basically we saw all that. We confirmed our hypothesis, but I will tell you, I will show them in more detail. So with these 21 different beetle taxa, we observed that the distance decay patterns in the north were flatter, but not only that, they were also quite similar. So the difference in the distance decay patterns came from the differences among taxa in the slope in the south. Okay, so the north flatter than the south, but also quite similar among groups. We also observed that this difference in the slopes between the north and the south was correlated with this richness gradient. So we were like quite in the track, or like this is going to be historical dispersal limitation, yes. And as you can see here, for carabus, which its wing legs. Most species do not have wings, so it's strongly dispersal limited. The slopes are stronger. These are just 2 examples to show you how the patterns look like. So we also tested if this difference in the distance decay pattern was actually related with those traits. And yes, it was. We didn't only relate it with dispersal traits, also with other ecological characters, but it was the dispersal ones, the one that explained most of the variance. So we were like, hey. Just a side note is that when we say we compared the distance decay patterns in the north in the south, we did not only compare the slope, we computed this index that takes into account both the intercept and the slope, because you can have same slope with different intercept, and that is actually quite different distance decay pattern. So with this study, we saw that the distance decay pattern in the north were relatively flat. It's probably only the good dispersers have reached those latitudes so far. That the dispersal attributes of the taxon explain the difference in the distance decay patterns. So we think this is clear evidence that historical dispersal limitation is a major determinant of biogeographic patterns in these groups, and we also think that this approach can be useful if it's applied to other taxa, for which we know even less. This is another study, and I will briefly explain how we have used this distance decay approach in the context of phylogenetic scaling below the species level. Okay, so it is as easy as if you have a phylogeny and you cut the tree to deeper levels each time, and you compute the distance decay of the turnover, the similarity in community compositions for each lineages, the distance decay that you can observe, and you can compare those curves. And why is that interesting? Well, this framework, we have developed this framework, and Andrés Baselga in one of his paper termed it multi-hierarchical macroecology, but it founded on the ideas that have been wonderfully explained by Belen in this book, that the processes acting at that there's some analogy between the processes acting at the genetic and at the species level, and this can be very helpful to understand what is going on in our biological community. So, in a very easy thought experiment, if you have a species and it's in a place and it's going to start expanding the range, what will we see? If it is only dispersal limited, there are only dispersal constraints, it will just spread like a water drop in all directions. Similarly, Andrés will talk a little bit later about this type of patterns that Adrian Castro has also studied, one of our PhD students. We have published a very nice paper with that. But species, the range of a species can be constrained also by niche processes. So in that case, we will not expect a circle, we'll expect something different, an ellipse, that the range will be deep limited. And as we are talking about large scale patterns, and the climate is structured in latitudinal bands, at this scale we'll be talking about this type of ellipses following these climatic bands. However, that's not the case at the haplotype level. If we study mitochondrial DNA, the mutual genetic parents, they are not selected. In this case, but they are within the species range. They are just limited by their ability to expand their ranges. Haplotypes, this mitochondrial nucleoplasm, you can test if they are behaving naturally or not. But if they are, and we have tested in our cases, they would be just expanded, right? So we can use this expansion at the haplotype level, the patterns that we will observe at the haplotype level that would be only driven by dispersal processes, with the ones that we observe at the species level that are potentially driven by dispersal processes or niche processes. So this is what we have done. So I think it's quite easy to see that the same way, if you have two sides, the same way as you compute the similarity of two communities at the species level. So how many species site 1 and site 2 share in this case? That will be only species B and C. In this example, the species are the letters and the haplotypes are the numbers. So you can do the same and compute how many haplotypes two communities have in common. So you can compute the distance decay pattern from biotic similarity at the haplotype level in the same way as you would do it at the species level. So we have done this type of assessment for two different groups in the same territory, in the Iberian Peninsula, that had multiple refugia. It's supposed to have had multiple refugia during the last ice age. So what we observe is that the distance decay pattern is the same, but with different intercepts. So the fact that the decay in biotic similarity is the same at the species level and at the haplotype level is an evidence that the same processes are acting at both levels. So it points to range expansion at the species level being largely restricted by dispersal processes. So to sum up, we think it can be very useful in this context of distance decay model to compare what happens at the haplotype level and use it as a benchmark to see if we observe the same pattern at the species level, because it's not necessarily the same. And if you, with our purpose, we have done simulations to show that you do not expect same slopes when niche processes are acting. And also that the evidence of the relevance of dispersal process would come as a fractal pattern with same slopes but different intercepts. And finally, I'm going to talk about distance decay in genetic structure. And as I have said, it's not distance decay, but because we actually see the increase, but it is the same type of analysis. You have to switch it. So the relationship between genetic distance and spatial distance within each species. So this is basically what we are doing is assessing an analogous pattern. What I have just explained about distance decay in assemblages in communities, we are going to translate it at the to genetic distance, right? So we will expect an increase in genetic distance with spatial distance. And in this case, we are interested in the shape of this relationship. Why? Because in the theoretical framework that was proposed by Hutchinson and Templeton, they clearly explained that we would expect a linear relationship between genetic and spatial distance in the case of good dispersor. Why? In the case of good dispersors, but in the case of weak dispersors, the relationship would be asymptotic, because we would observe gene flow due to dispersal when there is the increase in the curve, while when we reach the asymptotic, those are populations that are evolving independently, and it will be the result of gene-driven mutation. And if you think about places like the Iberian Peninsula with those multiple refugia where the populations have been isolated from one another and then the expansion from them, I think it's quite easy to see how this pattern can arise. So I said that we're interested in the shape, but also with the slope, because we can compare the relative dispersal ability of taxa depending on the slope. So a flatter slope would be indicative of weaker dispersal limitation, and a steeper slope would be indicative of stronger dispersal limitation. We did this for 3 different species of Hemalacus in the Iberian Peninsula, and we saw that in every case, the asymptotic relationship was the best support model pointing to this strong dispersal limitation. But not only that, the models were largely different in terms of slope and spline deviance, and this points to different dispersal ability among the species. So to sum up, with this study, we saw that historical dispersal limitation can be inferred from this asymptotic shape that it's analogous to how we are testing the functional shape in the distance decay models. And we also see that homologous are weak dispersers, but also, and this is relevant, that very closely related species can have very different dispersal abilities. So I think it has been quite long, but I hope today have shown you some interesting approaches that you can follow if you're interested in dispersal limitation or in distance decay approaches, like how we can kind of play with these patterns from a wider range of data that you may have at hand. And I have done this in collaboration with people here at the University of Santiago in the Natural History Museum, and I'm very grateful for everything I have done with all of them. Thank you very much.
Hello everyone, I am Sara Martínez-Santalla, and today I am going to tell you about a significance test for nonlinear distance decay models. Now I'm going to share my screen with you, and then we can start the presentation. So, there we go. As Carola told you before, distance decay of similarity patterns are very useful to answer a range of questions about biodiversity. However, many times, to be able to answer these questions and to make inference and to test hypotheses, we need to have a model of the relationship between similarity and distance. And the first step to fit a model is to know how this relationship is, how they are related. Nicole and Mahil in 2014 found out that the relationship between similarity and distance depended on the scale of data. If we have a larger scale data that allow for the turnover of species to happen smoothly, we can expect exponential distance decay functions. However, if our data is in a smaller scale, we are very likely to obtain power law functions. Then we have candidate functions for our model. We need to fit it. And we have chosen to do so using generalized linear models because they allow for zero value similarities, and this way we can avoid introducing bias by removing them from the data. The next step would be to know if the pattern that we are studying is present in the data and if we are capturing it with our model. This means that we need to test if the models are significant. This means that we want to contrast two hypotheses. Our null hypothesis would be the distance and similarity are independent, and thus there is no distance decay. This is represented in the picture with the blue model. And our alternative hypothesis would be that our data follow exponential or power law models. There are several tests adapted to test the significance of a GLM. However, we can't use them because they assume that data are independent for computing the p-value and the null distribution of the test. However, distance data in general are not independent because a distance or similarity matrix is constructed in such a way that every value in the matrix is the result of comparing two sampling points. This pseudoreplication effect can inflate type 1 error and power of a test that does not account for it. This means that it's going to give back false positives. So we need another way to estimate the distribution of the test under the null hypothesis. A common solution is to use the Mantel test instead to test the significance. What does the Mantel test do? Well, it's a correlation test that compares the correlation between similarity and distance in our data to what would be expected under the null hypothesis. And to estimate this null distribution, it uses row-column permutations to make distance and similarity independent without altering the structure of the data, this pair structure that I have just told you about. However, there is a problem with distance decay in our case, with distance decay, not with Mantel test in our case. Mantel test fits a linear relationship, and our models are not linear. This means that if we use the Mantel test, we wouldn't be really testing our models, but a linear one. The solution to pseudoreplication that we propose here, that would be able to deal with nonlinear models, is to adapt the classical tests, the classical significance test used for generalized linear models to distance decay data. These tests are based on the deviance of a model. The deviance of a model is a measure of how much it digresses from a saturated ideal model that fits all the data. And we can compare this deviance measure for the null model and for our fitted model and see if they are the same or if our fitted model fits better. This statistic takes the form of a pseudo R-squared, and now comes the big question. How can we estimate the null distribution of this statistic to avoid pseudo-replication? Well, we can import the idea from the Mantel test and use row-column permutations. And we went one step further and also tried another method, a block bootstrap, that incorporates this very same idea. So the objective of this work would be to assess the efficiency of a deviance-based significance test for exponential and power law distance decay models. And we want to do so in terms of type 1 error and power, and using row-column permutations or block bootstrap to obtain the distribution of tests under null hypothesis and correct p-values. To test the type 1 error, what we did was to simulate the null hypothesis of the pseudo R-squared statistic. This means that we had to simulate data where distance and similarity were independent. To do so, we first obtained species presence absence data from a binomial distribution and independently some coordinates. We then computed the Euclidean distance and the Simpson similarity, and we obtained data without distance decay, but paired. For testing the power, we had to simulate data under the alternative hypothesis of this test. This means that there is a nonlinear distance decay. And here I'm going to tell you about just one of the scenarios that we simulated. We simulated some more, but this is the most illustrative, and I have only included this one for reasons of brevity. So we obtain species presence absence data by using a Gaussian random field and transforming normal variables that it gives back to binomial. A Gaussian random field produces a series of sampling points, and in those points it produces observations from normal variables that in our case would equal species. And these normal variables in those different sampling points the values are going to be more similar if the sampling points are nearer in space, and more different if they are farther apart, following the covariance function that in our case was an exponential. We then computed the Euclidean distance and the Simpson similarity and obtained distance decay data. So Now I'm going to explain the row column permutations and the bootstrap. Both these methods rely on the idea of selecting all the values of a distance matrix that are related to one sampling point. For the permutations, first we obtain a permutation of the sampling points, and then we reordinate the similarity matrix accordingly. What this does is that makes distance and similarity independent of each other, thus simulating the null hypothesis of the test, but it still preserves the structure of the matrices, the spare structure. As for the bootstrap, the first step is to save all the values related to a single sample point in a block matrix, in that way that every column is a sampling point. We then resampled the blocks, sampling points, and computed their sample matrices, the same for distance and similarity. And now, to make distance and similarity independent, we randomize the values of the similarity or sample matrix by a column. This method duplicates the values of the resampling matrices. So we had to remove half of them. And to do so, we removed randomly from each column the same values in both matrices. Now that we have our sample or permutation data, we computed the statistic, and to estimate the distribution under the null hypothesis of the test, what we did was to repeat this procedure 1,000 times. So now that we have simulated data under both the null and the alternative hypothesis, we compute the value of the statistic. We obtain the distribution and the p-value of this statistic using row column permutations and block bootstrap. To estimate type 1 error, what we did was to repeat this procedure under the null hypothesis 1,000 times, and we expected to obtain something near to 0.05 value. For estimating the power of the test, we did exactly the same, but with the alternative hypothesis. And the results were that for the null hypothesis, for the estimation of type 1 error, if we combined the test with row column permutations under all the sample size, the different sample sizes, and for both the models, we were able to correct the effect of the pseudo-replication. The type 1 error was correct. The bootstrap did correct it too, except for the larger sample size, in which it was a bit high. So what we can affirm is that at least if we combine the test with permutations, we are able to correct the type 1 error, avoiding the effect of pseudo-replication. And for power, we have to look at the alternative hypothesis data. What we found is that no matter the model, no matter the sample size, both methods have the maximum power possible. They always detected the null hypothesis as false when it was false. So if we combine this with a correct type error, this means that our test is working. This procedure works. So to conclude, what we have learned is that our test works as a significant test for exponential and power law distance decay models in combination, at least, with row column permutations. And this means that it is a better option for nonlinear distance decay models than what is actually most common, that is using Mantel test. Thank you so much for your attention, and if you have any questions or any comments, you can write to my email And thank you very much again.
Hello, my name is Ramiro Martín-Devasa, and I'm going to talk about a new approach to study the distance decay pattern, the compare function. Now, I'm going to share a presentation. Okay. As explained before, the distance decay is normally studied by two functions, the exponential and power law function. These models have shown a very good performance fitting the pattern. They fit two parameters, an intercept, which is the similarity at the closest localities, and a slope, the rate of decay of similarity with distance. However, it can be some situations in which these models cannot be the better option. Let's see. It can happen that in communities with species with large distributions, the nearest sampling points can have the same species. In these cases, the similarity doesn't decay at the shortest distance and can start to decay at medium or large distance. In this scenario, the exponential function can have some problems. For example, it cannot adapt the initial plateau, and it can fit values over 1. This is a problem because the similarity indexes normally take values between 0 and 1. So a fitted value over 1 can lead to some misinterpretations of the pattern. The power law function have the same problem. It cannot adapt initial plateaus, and it can fit values over 1, leading to some misinterpretation. We think that in this situation, a sigmoidal function can be very useful because it can be bounded between 0 and 1, and it can adapt that initial plateaus. We propose the compare function. The compare function is a generalization of the exponential function, and is normally used to increasing patterns, like cellular division or bacterial growth. However, it can be easily adapted to a decay pattern. It also fits two parameters, an intercept and a slope. These two parameters have potentially can be interpreted as the intercept and the slope of the exponential or the power law function. The objectives of this work are to assess the influence of the species distribution size and the functional form of the distance decay pattern, and study the use of the compare function to fit the distance decay pattern. We also want to try to give the compared parameters a biological interpretation. To assess the influence of the species distribution size on the form of the distance decay, we first simulate some distance decay data. To do that, we simulate some species distribution as spatial, circular spatial polygons of different size. We simulate large distributions and small distributions. We also created sample points as spatial points distributed randomly. To have a present-absent matrix, we compute which polygons are overlapped in its sampling point. Then we increase the size of the small distribution, keeping the large distributions in a constant size. With this method, we can have different scenarios of distance decay with communities with small distributed species, small species distribution, and other with largest distributions. In both cases, we fit an exponential, power law, and compared function, and compare it with the Akeke criteria. Moreover, we repeat this same procedure, but keeping the distribution size constant and changing the proportion of large and small distributions. Then we have this the same case as in the previous with the previous procedure. We have different scenarios of distance decay, but this time in communities with different percentage of small distributed species and large distribution species with big distribution areas. We also fit an exponential, power law, and compare function, and compare it with the Akeke criteria. We also want to know how these functions works, not only with simulated data, but also with real data. So to do that, we use the 21 South European collector taxa. We use the data for the south of Europe because, as Carola has explained before, the European collector taxa shows a more variable distance decay form in the south than in the north of Europe. These different taxa have different distribution size and different biological characteristics that and also have different distribution dispersalability, you know? Different dispersalability that we can measure with some characteristic like the proportion after species or the mean size. We also fit exponential, power law, and compare function to this data and compare it with the Akeke criteria. Trying to give a biological interpretation to the compare parameters, we compute the correlation between the parameters and the biological characteristic of the coleoptera taxa. The simulation result shows that for the smallest distribution, for the scenarios with the smallest distribution and the less percentage of large distribution, is the exponential function who present a better fit. In the cases with the larger distributions, is the compare function who present a better fit. By increasing the percentage of large distributions, we can also see that it's the compare function who present the better fit. So the compare function present the better fit with the largest distributions and with the high percentage of largest distributions. The Beetle data, in the Beetle data, the compare function have a better fit than the exponential, the power law function, in 5 cases. The exponential function shows a better fit in only 2 cases. And in 14 cases, both models can be considered similar. In the correlation between the compare parameters and the biological characteristics of the Beetle taxa, we found a significantly correlation between the parameter B, controlling the intercept, and the mean size of the species. And also significantly correlation between the parameter C, the slope, and the percentage of apterous species. Both parameters have a significantly correlation with the range size, the distribution range size. So the Gomper parameters are significantly correlated with the biological characteristic of the beetle taxa. For conclusion, that the Gomper function have mathematically advantages because it's bounded between 0 and 1. So it can take values outside the range of the similarity indexes, and the exponential function and the probl function can fit values over 1. The Gomper function allows to accommodate initial plateau, the exponential and the probl can't, and the empirical data show that the Gomper function normally fits better or equal than the exponential function, and is rarely fits worse. And its parameters reflect the biological characteristic of the organism. So the parameters are interpretable in biological terms. With this, we recommend the use of the Gomper function to fit the distance decay data and study the distance decay pattern. Okay, and this is it. Thank you very much for listening.
Hello, I'm Andrés Baselga again. Thank you, you all, the previous speakers. I'm not going to talk about a different thing related to distance decay patterns, but as I said in my introduction, trying to find if we can measure the degree of anisotropy in community turnover and use it to learn new things about how biodiversity is distributed and how much species distributions are in equilibrium with climate. So most of you will know what isotropy or anisotropy is, but just very quickly, something is isotropic if it's uniform in all orientations, and anisotropic if it is not. If some, for example, a process occurs in with different strengths in different directions. So what does this mean? That isotropy is an absolute term. Something is isotropic if it's perfectly isotropic. As you see, as a macroecologist, I couldn't resist the opportunity to show the biggest scale possible. This is a map of the universe, and the universe is said to be isotropic even if there are tiny levels of anisotropy. And thanks to these levels of anisotropy, we are here. We can be doing this Humble Day celebration. But what I want to say here is just that anisotropy is a relative term. It can be the degree of anisotropy can be a tiny bit or a lot. In biogeography, isotropy has been studied on a very few occasions. I want to cite here this wonderful book about species distributions by Eduardo Rapoport. I recommend everybody starting in biogeography reading this book because it has a lot of very wonderful ideas. Something very interesting to read. Eduardo Rapoport realized that the shape of species distributions can tell us things about the processes that determine these species distributions and their limits. later, in their seminar paper about macroquality, Brown and Mother show these plots that relate the range of species distributions in east-west directions against the range in north-south direction. So each species is a dot, and if its range is elongated in north-south direction, it would appear above the diagonal, and if it is elongated in east-west direction, it would appear below the diagonal. As you will see, there are some deviations from the equality, that meaning that species are equally distributed in northeast or east-west directions, but deviations are not huge. But there is some degree of anisotropy at the species level. Some years later, we have assessed whether, no, I'm showing here just the amphibians, but we did it for many other groups of animals and plants in several continents. And the interesting thing here was the observed pattern was between the neutral prediction, which is just circular ranges with equal ranges in north-south and east-west direction. And as I said, the observed slope of this relationship was between the neutral prediction and the climatic niche prediction, as in many other occasions by the European macro. Why is this relevant for the token giving? It's just that we observe some levels of anisotropy at the species level, and this has to be consequences at the community level. Why? Because if species ranges are elongated in a specific direction, turnover will be faster in the other direction. So the anisotropy of species ranges would produce anisotropy in community turnover. Here is a very simplified toy model, just to show the intuition behind this relationship. If all ranges are circular, turnover would be equal in all directions, and if distribution ranges are elongated, turnover would be faster in the other direction. So why is this relevant? It is relevant because we know for sure that climatic gradients are highly anisotropic. There's nothing isotropic here. We have very well structured bands of, for example, of temperature from the equator to the poles. This is very well known, and this is why I think anisotropy is not assessed in macroecological studies, because we all assume the world is highly anisotropic. And why I think anisotropy could help? Because we already know that different taxa have different levels of equilibrium between species distributions and climate. Araujo and Pearson have introduced this variation across different taxa some time ago. Very recently, Lenoir and collaborators have shown that this degree of equilibrium between species ranges and climate is much higher in marine environments compared to terrestrial environment. So we have a huge variation in how well species and then communities would be able to track climate changes and which is well, the equilibrium between species distributions and climate are so important. I think anisotropy of community turnover can affect. So as we have already seen in the previous talks, distance decay models are usually implicitly modeled as an isotropic pattern, and with this model it as if the similarity, assemblage dissimilarity was uniform in all directions. So we just measure similarity between communities and then see if this similarity is correlated with the spatial distance or any climatic or environmental distance we're interested in. So then we build the model and just regressing linearly or non-linearly assemblage similarity against a spatial distance. This is a very common model. We have seen they are very useful to learn a lot of things about biological communities. So, as I said, we fit this model without regard to orientation, and this is the usual way to do it. And as I said, you can learn a lot of things from it. But I think if we look at orientation, we can learn even more things. Is that true that sides separated in different directions have the same similarity, and does similarity change with the same rate in all directions? We can test this, and we use 21 European beetle taxa, I mean super specific taxa like families and big genera. So we have 21 taxa with species presence absence matrices. We can we did that with European and American vertebrates, amphibians, mammals, and birds, and with marine phyto and zooplankton from Australian seas. And the method is very simple. We differentiate distance decay patterns for north-south distances against east-west distance. So what we do is just consider places separated in a specific angle or in a stripe of the geographical area, and if the pattern is isotropic, as I'm showing in the first row, both curves in north-south or east-west direction would be very similar, and if the pattern is anisotropic, both curves would be very different because turnover would be faster in one of the directions. Here, it's depicted in north-south direction. Turnover is much faster than east-west direction. So, what have we discovered with this? As predicted, in marine communities that are not, in principle, limited in the dispersal abilities, we see a huge anisotropy in which turnover is much faster in north-south direction compared to east-west direction, as we expect from the climatic rate. In terrestrial vertebrates, we observe anisotropy too, but it's not as marked as in marine environments. And we have an exception, which is European amphibians, in which the turnover is faster in east-west direction compared to north-south direction. And what happens in beetles? Beetles, of course, are very important, you know, because not only because I love them, just because they are a huge fraction of biodiversity. In beetles, and I'm showing here only a couple of examples. I will show the 21 different taxa later. The usual thing is to find a quite isotropic pattern with both curves almost identical, like in this example. And when we find anisotropy, as in the European amphibians, the turnover is faster in east-west direction compared to north-south direction. So here you have all the examples. We have like from ground beetles living in the ground, as the name say, longhorn beetles, leaf beetles, weevils, and carbon beetles. A lot of different examples, and there are two very interesting things here. A lot of taxa have isotropic pattern, and when they are anisotropic, in most of them, turnover is faster in east-west direction, contrary to the climatic breading, which is much marked in north-south direction. And there is a huge variation in the degree of anisotropy, meaning that probably different characteristics like associated to dispersal or niches make the anisotropy be different in different beetle taxa. This is very interesting, as Carola has introduced in her first talk, because we have a lot of different beetle groups, so we have statistical power to compare why anisotropy changes from one beetle group to another beetle group. And we have assessed that, and what we have seen is that the traits correlated with dispersal ability, like the proportion of wingless species in each group, is very tightly correlated with the degree of anisotropy measured just like the difference in slopes between east-west and north-south direction. And in turn, the niche attributes like habitat type is the only trait, the only niche characteristic that we found is significantly correlated with the degree of anisotropy, explain much less variance, and it is this variance is nested within the dispersal traits. So, in conclusion, anisotropy varies widely across taxa and systems like marine terrestrial systems and continents. In Europe, the peninsulas in the south make the turnover in east-west directions be much important than the turnover in north-south direction. Anisotropy seems related to dispersal ability more than to niche characteristics, and this suggests that anisotropy would be a good measure to infer the degree of equilibrium between species distributions and climate. And this, of course, is super relevant to predict the effects of climate change on biodiversity and on biological assemblages. So I finish here. Thank you for listening to this talk and for attending the symposium. We hope you enjoyed it. And we thank again the organizers of the International Humboldt Day, the IBS, and goodbye.